So people can see this?
Yep.
All right, cool. So I'll describe something about neurofeedback and my study. I just want to say that this is something I'm going to present to my PhD defense. So any sort of comments and critique and, you know, I didn't understand this or you went too fast here, and ask also if I'm too fast with something, then please sort of stop me and ask me to go back. Yeah, any comments is helpful for me, even critique. So yeah. So what I'll go through is the difference between correlations and interventions, which is very relevant for neurofeedback. And I'll describe neurofeedback and operant conditioning of brain activity. And I'll describe some examples for basic science and clinical science. And then I'll go into multisensory perception, which is where I performed a study using neurofeedback training of multisensory perception. And lastly, I'll discuss some perspectives. So, but actually, the thing that I investigated most in my PhD is the relationship between shark attacks and ice cream sales. So we all know that there's a tight relation between shark attacks and ice cream sales. Whenever shark attacks increase, ice cream sales also increase. And whenever ice cream sales decrease, shark attacks also decrease. So I just want to ask, are there anyone who can explain to me why there is a relation between these two? Any ideas? I'm already giving you a hint here that there might be some underlying cause that's affecting both shark attacks as they increase and ice cream sales increase. What could it be? Come on. You know it. Anyone? Okay. This is easy. Come on. It's temperature. It's when the sun is out and there's more sunlight, people tend to go to the beach. And as they do, there's more shark attacks. And they also tend to buy more ice cream. And there's no causal relation, meaning it's not as if as you eat more ice cream that there's more shark attacks, or that there's more shark attacks, now you suddenly eat more ice cream. And this problem is the same when you're dealing with a cognitive phenomena like perception and neural activity. You have some observed neural activity and some observed cognitive phenomena, but you don't know if there's an underlying neural activity which is actually causing both the cognitive phenomena and the observed activity that you're seeing. So in order to gain confidence that this observed activity is really important, what you need to do is you need to modulate or control the intact human brain. You need to modulate this. And as you do, you need to increase this activity or decrease it, and see a change in the cognitive phenomena, increase it or decrease it. So improve perception by improving some activity in the brain. And that will allow you to exclude the possibility that this activity is important. So this becomes unimportant, and this shows you that this is the underlying activity related to the cognitive phenomena. So how do we do that? We use interventional techniques like TDCS and TMS, where you apply an electrical current to the brain. But another option is neurofeedback, where you measure the brain activity in real time and show it back to the participant such that they gain control over their brain activity, and you influence in this activity here. So you could say, I'm going to explain this further in neurofeedback. You could say that these techniques are exogenous stimulation because they apply a current from outside the brain, whereas neurofeedback is an endogenous stimulation. This is also self-stimulation, although that has some different connotation. So endogenous stimulation is from within, and here neurofeedback is just a different type of stimulation. So I'll just quickly go over that there are different ways of measuring the brain. We have MEC and EEG, which measure brain waves, and I'll primarily be discussing these. And then we have fMRI, which measures bulb response, although brain waves are linked with the signal you're getting from fMRI, that the brain waves are linked with metabolism. There's some differences in resolution, but generally they measure the same thing. And brainwaves are also linked with neuromodulators like serotonin and dopamine. So even though we have very different techniques, they sort of measure the same thing in some way. So neurofeedback is a psychophysiological procedure in which online feedback of neural activation is provided to the participant for the purpose of self-regulation. So you have a test subject and you have a brain scanner where you pick up a signal. You then measure it fast enough such that you can show it back to them and they gain control over the brain activity. This was first started in the 1950s when it became very interesting to look at these expert meditators who were able to perform extraordinary feats of self-control by not perceiving pain as painful, by this yogi here sitting on a bed of nails, and it became very interesting to see at their brain activity what they were doing in order to control their pain. Then came Kamiya, who performed experiments to show that you could downregulate alpha activity in order to reduce anxiety. This was published in Science. And then at around the same time, Barry Sterman figured out, and this is NASA-funded research, that if you gave rocket fuel to cats, at some point they would vomit and they would salivate and then they would have a seizure. But if cats had their alpha activity conditioned, meaning that they had trained to increase the alpha activity, they didn't have the seizure. So you could train cats to avoid having a seizure from rocket fuel. This is what NASA were doing in the 60s. And this eventually became something that could be used for epilepsy. So time for more cats. What are we seeing here? This is my favorite video in all the internet. So I don't know what you guys are seeing, but if you're a normal human being, what you're seeing is an amazing, funny video. But if you are a psychologist or an abnormal human being like me, what you're seeing is a discriminatory stimulus and a behavioral response from the animal. And that response leads to a reinforcing reward. And operant conditioning states that physiological responses increase in probability if they are reinforced in the presence of the same discriminatory stimulus. So basically, in layman's terms, the cat will repeat the behavior because it's being rewarded for doing it. Now, what if you didn't have to have the cat perform the behavior, but just have the physiological response in the brain? So as the cat is imagining pressing the bell, we record that activity and show it back to the animal, or just give it a reward for having that activity, we would be able to reinforce a physiological response rather than a behavioral response. And this has actually been shown that even individual neurons can do this. So individual neurons, if you give them dopamine when they fire at a specific frequency, they increase the firing rate when they're given dopamine for that activity. So this is a principle which is prevalent throughout the nervous system, down to even the one single neuron. And this is operant conditioning. And this is what neurofeedback relies on. You're measuring some brain activity fast enough to show it back, and you're reinforcing that brain activity. So this has all manner of applications. You can take a hard to detect stimuli, like here, and measure what is it that's difficult in terms of detecting the stimuli, figure out when do participants see the stimuli versus not see the stimuli. And you already discussed this in Christian Sandberg's lecture, where he discussed this type of paradigm where you either see a stimulus or don't see a stimulus. And you can train the activity related to seeing the stimulus such that participants see the stimulus more often. Or you can, in touch perception, figure out this is somatosensory cortex. If you increase alpha activity in the somatosensory cortex, participants can become better at a touch perception task. This has obvious clinical benefits. So for example, within tinnitus, you can measure the brain activity associated with tinnitus. Tinnitus is you're experiencing these loud auditory sounds. Then you downregulate the activity in the brain related to tinnitus, and participants have lower distress in terms of tinnitus. And it's better than other treatment options for tinnitus, like providing TMS to the brain and providing something called frequency discrimination training, which is a type of therapy for tinnitus. Neurofeedback outperforms them, although the effect is short-lasting. It's gone after 6 months. Then there's pain perception, where it was shown in 2005 that you could control the brain activity related to pain perception. And this was compared against 4 different types of control groups, like imagining the pain was lower, attending to the pain, getting feedback from a different brain region than what was related to pain, and receiving feedback from another person's brain, which will not work. It needs to be your own brain that you're receiving feedback from. And the neurofeedback group outperformed them. And this has actually been replicated in some recent high quality studies. So what neurofeedback allows you to do is to look at correlational evidence and to say, we can show that gamma activity, for example, is related to perceptual awareness. These are some studies performed where they show that gamma activity is related to perceptual awareness. And then we can go and say, well, what if we ask participants to increase their gamma activity? And here it shows that they're able to do so. This is before neurofeedback, and then after neurofeedback, they increase their gamma activity. And now the neurofeedback group, this group here, detects these objects more than the control group. So we can take correlational evidence, gamma activity is related to perceptual awareness. We intervene on gamma and see that awareness increases. This is also, so taking from what Lau has just talked about, you can do this with decoding too. So you can figure out what in the brain is related to perceiving these degrees of lines, or these degrees, or this degree here, and have an induction of that brain activity related to the perception of this, or that. And if participants are given a task where they're supposed to discriminate between these, they improve on the activity that you've trained with neurofeedback. So you can use decoding measures, combine it with neurofeedback to train very specific cognitive functions related down to even orientation of lines like this. So the clinical effects are what interest me the most, and tinnitus is my sort of prime example where I feel that I could confidently say that neurofeedback works. It's been tested against other types of interventions, and it's not a question of whether it works, it's just how do we get it to work better. So I will approve it for use, you could say. But then there are other areas where it's more difficult, like for example, ADHD. There's a big question about whether neurofeedback works for ADHD. There's been one study that I really like, which compared neurofeedback to EMG biofeedback. So that's where people with ADHD just learn to relax their shoulders and learn to relax their muscles. And here the two groups show similar behavioral improvements on ADHD symptoms. So I will not say that neurofeedback is better than other approaches. And there's also been some disagreements in meta-analyses about whether ADHD is really relieved following neurofeedback. But here I just want to say that there's lots of new studies. There are some new studies showing that there could be effects, but I'm going to say that it's not ready for use here. And this leads me to what I wanted to sort of investigate with my PhD, and which is visual spatial neglect. Or rather, I should say that I wanted to think about how I could perform such a study in spatial neglect. So visual spatial neglect is often occurs after brain injury to the right parietal cortex, where afterwards you are not able to allocate attention to the left half of your visual field. So this is a truly debilitating mental or brain injury. Patients can't see one half of their world, basically. They often don't put makeup on that side, or they don't eat that side of the plate. And this means that in a task where you are seeing lines like this, and you have to dot over the lines and draw over the lines, they're not able to see these lines, so they're not able to cross them. And this is called omissions. If you downregulate alpha activity using neurofeedback, the level of omissions decreases. However, this is some results in very low sample studies, and they are not compared against any control group. So that's why I would say that even though there's some indication it could work, it's not really ready for clinical use. So this is the point in the talk where I would love to explain that I had performed an experiment in patients, in visual spatial patients, but this is quite hard to do neurofeedback experiments. And therefore, I reasoned for my PhD that I would have to work with healthy volunteers first, using a convenient sample, because patients are hard to recruit. And it's also the case that for healthy volunteers, things can go wrong and you can learn how to perform this procedure, whereas in clinical research, it's hard for things to go wrong. So my idea was, if I can train healthy volunteers to perceive the world differently, can I train patients to perceive this left half of the visual field? So what I wanted to investigate was this multisensory phenomena, a sound-induced flash illusion. And this relates somewhat to what Janice also talked about multisensory integration a couple of days ago. So in this illusion, what you're seeing is you're just watching this flash appear, and you have to judge how many flashes appear. In actuality, there's often only one flash. However, there are two sounds, two beep sounds, and they come really fast. So you have a flash and a beep, and then a second beep. So it says beep, beep, but only one flash. And what happens for most is that half of the time, they actually perceive two flashes rather than one. This leads to the idea that what you see is what you hear. And this is because for vision, when you want to estimate things in spatially, when you want to say, is something here or here or here? Is there something you want to see? Is this over here or over here? Vision is very good for doing that, but that's not as good for auditory information. So let's say you had something that was 100 miles away and you needed to, or 100 meters away, and you needed to discriminate whether a tree was to the left or to the right of another tree. Your vision is very much able to discriminate those two. But let's say instead you had two sounds appear 100 meters away. Your auditory sense is not able to pick up on that difference. But when it comes to temporal information, audition is better. So if I, for example, present 4 flashes really fast, like flash, 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 maybe that was 5, you're not able to see it. It just comes so fast. But if I say beep, 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 what you're able to do is you're able to say 1, 2, 3, 4, 5. You have a better temporal understanding of the beeps, even if they come fast, because audition just has better temporal resolution. And this means that when you're presented with a flash like this and 2 beeps like this, you are more relying on audition to make this. And participants actually perceive an additional flash. Like there is a real flash they perceive. It's something you have to experience in order to truly understand it, but they do see an additional flash. So more flashes, when you have more flashes, you have less alpha activity in the occipital part of the brain, the posterior part, the parietal and occipital areas, whereas more alpha leads to less flashes perceived. But here we have the problem again, that the observed alpha activity that we're measuring is related to the cognitive phenomena, but we don't know if there's some underlying activity that's causing both alpha and the cognitive phenomena. So what we need to do is intervene on the alpha activity and see that the cognitive phenomena changes without a change in some underlying activity. So this is what I did for my PhD. I had participants come in and I presented these flashes in either the left side or the right side while I presented these auditory beeps so that they experienced this sound-induced flash illusion. And I measured them both in the left side and the right side. And initially, this is sort of hypothesized results, but this is also correct that most of them experienced the illusion mostly in the left side and the right side. Then what I could do is allocate them to two different groups. So one group, the left neurofeedback group, downregulated alpha in the right side and upregulated alpha in the left side, whereas the right neurofeedback group got the opposite. So I'm biasing alpha to be more in one side than the other. And every time alpha goes down for this group and this goes up, this visual stimulus here becomes more clear. And I've asked my participants to make this more clear. So they're receiving neurofeedback related to their alpha activity in this parietal area of the brain. And what I then hypothesize is that afterwards, in the post-test, they should experience more illusions in the side where alpha was trained down, according to where it's presented and how the information in the brain crisscrosses over into the other hemisphere. So what I need to say here is that I had three different types of illusion conditions where there were 2 beeps and 1 flash, 3 beeps and 1 flash, 4 beeps and 1 flash. And then I also had control conditions where there was just 1 beep and 1 flash, 2 beeps and 2 flashes, so that participants didn't always know that there were more beeps than flashes, and that there actually were some trials where there were 2 flashes, 2 real flashes. So this is the real-time magnetoencephalography setup that I created in Skyview. So this is the scanner, and then I'll go quickly over these technical things here. An acquisition computer gets the data, sends it to a classification computer, which performs all the hard work, figures out if alpha is higher or lower, sends that to a stimulus computer, which provides it back to the participant such that they gain control. And this is the setup here. The participant is inside the scanner and it's sent over to the acquisition computer. Then that's sent to the classification computer and further on to the stimulus, and then back to a projector, which sends the neurofeedback back to the participants, thus completing the loop. And in reality, this is what it looks like. So this is the stimulus computer, and this is my classification computer doing all the hard work. And over here, we have the participant in there. And here we have the acquisition. So this is all the data that's coming in, the MEC signals from a lot of different sensors. And this is a script that I'm running, just a Linux script, which sends the information onto the network. And here we have, this is a very important cable for the study to work. It's getting the data. And now it's acquiring data about what the participant's brain is like and how much alpha is in one hemisphere compared to the other. And I'm sending that information back so that it's changing this visual display. And I'm also acquiring eye tracking data here. And the loop takes some 180 milliseconds. So there's a 100 millisecond delay between what alpha is in the brain and as the participant is seeing. So it needs to be as fast as possible. I'm just going to go quickly over this. This is what Lau also talked about. In order to evaluate whether they change their brain activity, we perform a type of analysis called source spacing, which involve using the MR signal along with the MEC signal to sort of figure out where this came from. And this is called source spacing. And Lau was actually very helpful in helping me learn this. This is something that can be learned in a couple of months. It took me maybe 3-4 months to learn. So the results show that the most promising results were with the left neurofeedback group. So when I compared left-sided versus right-sided alpha, this is during neurofeedback, are they able to increase their left-sided alpha and decrease their right-sided alpha? And this is what they were able to do. Alpha was increased in that hemisphere compared to the other. However, for the right-sided group, the effect was not present. So it's perhaps more difficult to to increase right-sided alpha compared to left-sided alpha. And this is actually something that has also been shown in other studies, that increasing right side compared to left side is just more difficult, and we don't know why, not yet. Then what also happened was, because I'm presenting these flashes, I can compare how strong of an evoked response am I getting in that area that's processing it. And here it's shown that I am getting a stronger response for the left group compared to the right group in this right-sided hemisphere. However, there is no stronger response in the left side, in the left-sided hemisphere. So the most important part is, of course, behavior. And here I had these 3 illusion conditions: 2 beeps and 1 flash, 3 beeps and 1 flash, 4 beeps and 1 flash. And I, at first, did not see any effects on behavior. So there was no difference between the trained hemisphere and the untrained hemisphere. They were the same. And I would have hoped that the yellow bar here was up here and the green bar was down here, but that's not what I saw. Then, as I interrogated the data, I found that during the first 5 minutes following neurofeedback, there was actually somewhat of a difference. This was on the 2 beep and 1 flash. So it seemed as if there was sort of a short-lived effect, you could say. And it surprised me that it was this condition here. And I didn't understand why it was only this condition and not the others. But as I started to think about it, the 2 beeps and 1 flash condition is, auditory and visual signals are, you could say, more likely to originate from the same source, because the difference is very low between 2 and 1 flash. Whereas for example, 4 beeps and 1 flash, the auditory and visual signals are more unlikely to originate from the same source. Or you could say the brain it's less that the brain wants to infer that the 2 things originate from the same source because they are so different from each other, the 2 signals. Whereas here the signals are more similar, the brain is more likely to interpret that they come from the same source or event in the world. And this is perhaps what alpha is doing. Maybe alpha is not involved in seeing more flashes per se, but involved in resolving the inference, in helping in the brain to infer that the signals come from the same source. So the brain is inferring that the 2 signals likely originate from the same event in the world. Or in another way, you could say that multisensory integration breaks down when the disparity between the signals becomes too large. This is what I started to sort of think about. And then working on this idea that maybe alpha activity is not causing the cognitive phenomena per se, but involves an inferred environment or an inferred causal structure between the auditory and visual signals. And this is actually, I put this here because this is actually similar to what Janice talked about a couple of days ago with oral referral, that when you're to estimate localization of smell and taste, those 2 senses merge more together when sucrose, for example, is paired with vanilla, because you've already built up this idea that the 2 things come from the same source, from the same thing in the world, whereas water and vanilla don't relate, and caffeine and vanilla don't relate, but sucrose does. So the brain is inferring that the 2 things originate from the same thing, and maybe that's where it is. So then I read this study, again, a study I should have read more carefully, perhaps, before I performed the experiment, which is a very cool study. And here they actually showed that the previous trial, so the trial occurring before any trial in the study, had an influence on whether or not people perceived flashes or not. And it did so relative to this disparity. So when you have 2 beeps and 1 flash, the effect of the previous trial was larger. And they also showed that this was related to alpha activity. So this sparked an idea for me that maybe I should be looking instead at whether, for example, this is a control condition. So I had 2 beeps and 1 flash, or 2 flashes. So this is a place where there's just been a control condition where the two signals really look as if they come from the same source, because I have two flashes and two beeps. And then I could be looking at this 2-beep 1-flash trial and see if that influences it. Whereas, for example, in this case, you have very low previous trial numeric congruency. You have it so that it's very unlikely that the two signals come from the same source, and then afterwards I'm measuring the same thing. And this is where I found that it is actually here that I'm seeing my effect. I'm seeing that in the trained hemifield, multisensory integration increases for that trained hemifield only when the previous trial is highly congruent and the next trial has low numeric disparity. Whereas if the previous trial has low numeric, previous trial numeric disparity, then there's no effect. So what I'm able to write about is then that the neurofeedback modulation of the sound-induced flash solution using these parietal cortex alpha oscillations, they really depend on the previous trial and the previous multisensory congruency. So we find that training effects were present for illusion trials with the lowest numeric disparity, 2 beeps and 1 flash, only if the previous trial had high congruency, 2 beeps and 2 flashes. And you could say that the present results show that low alpha in these posterior regions, they bias observers towards multisensory integration when they have learned that the audiovisual signals come from the same source or common origin. And this is consistent with Bayesian causal inference and a new type of theoretical account of multisensory integration. So perspectives. These studies, they need this study and these results, they need to be replicated before we're certain about this, because it's a very small study, and what we weren't designing our study to find this effect. This was something that I sort of ad hoc added to my investigation. But the study does present a new way to investigate multisensory perception. And it shows that the previous trial is important. And this is something that I think the rest of neuroscience maybe should start to sort of also look at, is how is the brain adapting to the previous trial as it's about to see the next trial. I think that this is something that will become more and more important in the next 10 years, 20 years within neuroscience. It shows that the brain is inferring the environment, and that this is very important. And these results are actually predicted by a couple of review papers that actually predicts my results, or spot on. And then there is a clinical perspective here, because we can decrease alpha in the right hemifield, or this you say hemisphere. We can, and we can increase visual responses in the right hemisphere. As I showed, alpha could be downregulated in the right side or increased in the left. And this led to these stronger visual responses. And that might actually be what a person with neglect needs, to have more sort of energy in that hemisphere, and then be able to perceive whatever visual stimulus is coming in. And lastly, I just want to say some sort of personal thing about it. I think I had this idea about neurofeedback that either it was going to sort of resolve all the problems, sort of treat every mental disorder in the world, or I also had this idea that maybe neurofeedback didn't work at all and was just completely useless. Whereas after performing the study, I've adopted this type of what I call optimistic skepticism. I think neurofeedback can work, although I'm very skeptic about results and sort of how it's performed, and I think everyone should be. But the thing about it is, neurofeedback works using operant conditioning, and operant conditioning works. It's prevalent in psychology. It's down to even a singular neuron can be conditioned using reward. So of course it is going to work, but it's not yet at the point where we can take any mental disorder, look at that brain activity, and affected so that it provides therapeutic, sort of meaningful therapeutic benefits. It's not a panacea. It's not a solution or remedy for all difficulties and diseases yet. That's what I want to say. Yep. Thank you.
Thank you, Timo. That was great, man. I will turn off the recording now, and so people can turn their cameras back on.